Hello and welcome to the pH and buffers lab. Today we're going to be looking at three unknown solutions, looking to identify these as either an acid, a base, or a salt. And then we'll also be comparing the buffering capacity of pure water versus a buffer called tris buffer. And we'll be measuring all of these things with a pH meter, which we will start off here by calibrating. So we're going to start off here by calibrating our pH meter. But first, we're going to need to label the glassware that we're going to be using for this portion of our experiment. So I'm going to rip off a small piece of label tape, place it onto the beaker. Then I'm going to do that for the other one. Place it onto the other beaker. Then I'm going to label them with the solutions they're going to have inside of them. So this one here is going to have our pH 4 buffer. So I will mark on the side here, pH 4 buffer. This one's going to have the pH 7 buffer, so I'll label this one for the pH 7 buffer. Now that my glassware is labeled so that in case I walk away and come back and forget which one was which, I know which one is which, I'm going to go ahead and pour in some of our pH buffers into our beakers. I only need enough to put the electrode into the beaker, so I'm going to pour just a little bit and then we'll see if this is enough, and if we need to, we can add more later. But that way we don't waste any unnecessary buffer. So now I'm going to take these, put these to the side, and I'm going to get out our pH meter. So it's in this plastic box here, sealed with tape to let us know that it's been cleaned. I'm going to remove the tape from the exterior of the container and open it up. Now, the pH meter has this helpful reminder here to not spill. The base here of the pH meter has some buffers in it that keep the electrodes working in between uses. So you want to be careful when you're taking off the pH lid. And sometimes you have to tear the parafilm just a little bit to get it out. So I'm going to make sure that I put this off to the side here, standing up. So now I'm ready to calibrate my pH meter. So I'm going to press the on button here, which is going to turn the pH meter on. I see that the display turns on. I am now going to calibrate the pH meter using our buffers. So I'm going to go ahead and hit the calibrate button here. And now it's ready to be submerged in our solution. So it actually looks like I'm going to need a little bit more of the pH 4 buffer here. So I'm going to go ahead and put that in. So now we'll submerge the tip. And there's two numbers on the pH meter. The top number is going to be the last calibration number, and the bottom number is what the current pH is reading. So right now, the calibration number is saying that this is a pH of 5, and the current reading is currently calibrating to get to the right pH value. So we'll just wait a little bit until both numbers are the same, and we're looking for both to be a pH of 4. So now the pH meter has finished calibrating, so I'm going to hit the hold end button here. And now I need to rinse the pH meter to perform the calibration at pH 7. So I'm going to take the pH meter here and this DI water and rinse it into this waste beaker. Then I'll take a Kim wipe and pat the pH meter dry. I want to be very gentle so that I do not damage the electrode at the bottom. Once the pH meter is dry, I'm ready to calibrate it with my pH 7. So I'm going to take my pH 4 buffer and move it out of the way since I no longer need it. Now I'm going to repeat the same process. I'm going to place the pH meter in my pH 7 solution and hit the calibrate button. So now we will wait until the pH meter reads 7, and then we will then be able to start conducting our experiment once our pH meter is properly calibrated. So now that it's finished at pH 7, I'm going to hit the calibrate button again. And now it's saying that the pH in the solution is around pH of 7. There's always going to be a little bit of noise and variance in the measurement, but it's within acceptable parameters of only about 0.01 pH. So now that our pH is properly calibrated, we're going to rinse it one last time to our waste beaker here. Then we'll get another Kim wipe. Again, making sure not to damage the electrode. We'll gently pat it dry. And now we're ready to begin the next step of our experiment. In this portion of the experiment, we're going to pour out our fluids that we'll be using for our actual experiment. We're going to add each of our unknown solutions to a small beaker so that when we get those solutions to add them to our experiment, we don't contaminate our stock solutions. So we're going to take slightly more than we'll need for the experiment, put them into these beakers, and then put our stock solutions back into our stock. Then we'll also be adding the water, which is going to be the essentially the reaction solution for our first portion of the experiment. So let's go ahead and add each of these solutions to these beakers. So we're going to want to add around 15 milliliters. Now, we're only going to be using 5 in our reaction. So as long as we add more than 5, but not too much more than 5, we don't really need to worry about how much we're adding. So I'm just going to eyeball about 15 milliliters. So now that I have solutions A, B, and C, I'm going to go ahead and add the water. Now the water we're adding, the amount of the water is important. So we can't just eyeball the 20 milliliters that we need. Instead, we need to use this graduated cylinder here. So I'm going to use my DI water spray bottle to add 20 milliliters of water into this cylinder. So while adding the water, I want to make sure to avoid putting any droplets onto the sides. And then when I measure the water, I want to come down and measure where the meniscus is. So now I'll take our 20 milliliters of water and add it to our reaction vessel. Now if you notice on this vessel here, I've labeled it as DI water plus solutions A, B, and C. That's because eventually all three solutions and the DI water will be added to this class. I want to make sure that at any point in the experiment, everyone knows what could be in there rather than just what might be in there at that given time. The next step is going to involve us adding our three unknown solutions and measuring the pH. So our first step is going to be measuring the pH of our pure DI water. So I'm going to put our pH meter into the water and allow it to stabilize. So now here we can see the pH of our pure water. So now we'll clean the pH meter and add our first unknown solution. So just like we were doing with the calibration, we'll take our waste beaker, rinse off the solution with DI water, and then gently pat it clean with a Kim wipe. Now, obviously here we're washing DI water off with DI water, but it's still important to repeat every process that we do to each of our experiments in each of our controls. So, since we're going to be doing this washing process after the addition of each of these solutions, we want to make sure we do it with our pure DI water control as well. So now we're going to add our first unknown solution A. So I'm going to take my glass transfer pipette and put it into our suction device here, our pipette pump. So I want to put it in so that it's flush with the back here. As soon as I start to feel a little bit of resistance, I don't want to stop. I don't want to keep pushing because then I could risk damaging the pipette, the pump, or myself. So now I'm going to take up 5 milliliters from this transfer pipette. So I'm going to rotate it so that I can hold it comfortably and still read the demarcations on the side here. So now I will slowly draw up 5 milliliters of solution A. What's nice about these is that I can be very exact with the volume that I'm moving. So now that I've taken up my 5 milliliters, I'm going to go ahead and deposit it into my reaction vessel. Now, I'll take my voided pipette and put it into my waste container. Now I'm ready to measure the pH of my DI water and my unknown solution A. So now here is the pH of our mixture of the DI water and our unknown solution A. So now that we made our pH measurement, we're going to go ahead and wash our pH meter again, and then we'll add our unknown solution B. So now that we've added A, I'm going to go ahead and move this off to the side. Now we're going to want to add the same volume of unknown solution B to our experimental mixture. So once again, I'll load into the pipette. And I'll draw up 5 milliliters of unknown solution B. Then I will add it to our experimental mixture. Now I'll take the voided pipette and place it into our waste bucket. Now we'll measure the pH of our solution with B. So here's the pH of our DI water and solutions A and B added to it. Now that we've measured our pH, we're going to clean our pH meter another time. Now we're going to add our final unknown solution, solution C, to our experimental mixture. So once again, take the pipette tip, put it into our pipette pump, and then I'll draw up 5 milliliters of our unknown solution. So I accidentally sucked up an air bubble, so I'm just going to void it and then redraw up the volume. This is one of the reasons why we like to make sure that we don't use our stock solution. Because now, worst case scenario, if I've contaminated anything by having to redraw the solution, I've only contaminated this excess amount that we've put into this beaker and not our whole stock. So now I'm going to go ahead and add this to our reaction vessel. Now I'm going to take the voided pipette and place it into the waste bucket here. And now we're ready to measure the pH of our final mixture. And so here's the pH of our final mixture of solutions A, B, and C and DI water. So now we're going to check the effects of using a real buffer and how that will affect the changes in terms of magnitude that we see in pH. So the first step is going to be measuring the pH of our new tris buffer. So here is the initial pH of our tris buffer solution. So now what we're going to do is we're going to do what we just did with our DI water, but now we have our tris buffer solution. So, just like before, I'll use the pipette to transfer 5 mils of unknown solution A to our experimental vessel. Then take our pipette, put it into our waste. And now we'll measure the pH of our tris and solution A mixture. So here's the pH of our tris and unknown solution A mixture. So now we're going to add 5 milliliters of unknown solution B to our reaction mixture. So again, we'll use the pipette to draw up 5 mils. And we'll void it into our mixture. And dispose of our pipette. and we'll measure the new pH. So here's the pH of our tris buffer and a mixture of unknown solutions A and B. So now we're going to add our final solution to our experimental mixture. And we'll dispose of our pipette and measure the pH. And so here's the pH of our final mixture of our tris buffer and unknown solutions A, B, and C.